Okay, so this is about where we left off. We were talking about photosynthesis. I'm sorry, not photosynthesis. We were talking about mitosis. And please remember the important definition of mitosis, and this is purely division of the nucleus, right? It's not cell division, it's just division of the nucleus. But we were talking about the division of the nucleus, mitosis, in the broader context of the cell cycle. Because each one of our somatic cells, and that is a body cell, is going to go through a natural process of reproduction. And it's going to divide to produce two daughter cells that are genetically identical to that mother cell. We just very quickly mentioned the sister process to mitosis, and that's meiosis. And remember, meiosis is the nuclear division process that we use to create gametes. And so this doesn't occur in somatic cells. This only occurs in gametic cells, in tissues that produce gametes. So we can look at the cell cycle that's associated with mitosis and break it into three distinct phases. And we need to say distinct in air quotes because they're not really distinct. But for the sake of simplicity, we treat them as such. So the first phase is something called interphase. An interphase is kind of a preparatory phase where all the things that a cell needs to do in order to divide, it accomplishes during that time. So it's going to be replicating organelles, it's going to be replicating proteins, it's going to be replicating its DNA, and getting ready to ultimately divide. So interphase is broken into three phases as well. There is a gap 1 phase, a G1 phase, in which those preparations are begun. So organelles will begin replicating, or the cell will begin replicating its organelles. Important proteins in the cytosol will begin increasing in concentration. So that ultimately, right, when the cell divides, those two daughter cells have all the things that they need to survive autonomously. After the G1 phase, there is the S phase, or the synthesis phase, in which DNA is replicated. So we go from a chromosome that has just one copy of all of its DNA to chromosomes that have two copies of all of their DNA. So those two copies can be segregated into daughter nuclei. Then we have a G2 phase where all the final preparations are made. Excuse me. So this is where organelles with their own DNA, things like mitochondria and chloroplasts, replicate themselves. All the organelles are completed. And then we enter the mitotic phase, which is the second phase in the cell cycle. And the mitotic phase, or mitosis, is the process of nuclear division. So the nucleus will divide. And then after a cell has two nuclei, then the cell divides. And that's a process called cytokinesis. That's called the C phase. Interestingly, after the C phase completes, the cell goes into what's called arrested development, and it won't divide again until it receives something called a growth factor that says, okay, it's time to start growing and dividing again. But it's an important thing to remember that the default position for all cells is to not divide. And that's an important observation in the context of cancers that we'll talk about a little bit later.
I have a quick question.
Okay.
What'd you say happens after C's phase? What the cell goes into?
The cell goes into arrested development.
Okay.
Yeah. Which is a great show, too, if you like those kind of shows. Okay. So let's talk a little bit about, well, let's define a few terms that we're going to need for this lecture, meiosis, and then also ultimately genetics. So there is a variable number of chromosomes across organisms. Our species has 46 chromosomes, right? And other species have some variable number. And we used to think that there was like some correlation between chromosome number and how evolutionarily advanced a species was. And we understand now that that correlation just doesn't really exist. Organisms just have different numbers of chromosomes. But those chromosomes, our 46 chromosomes, are really distributed or organized into 23 pairs of homologous chromosomes. Now, what a homologous chromosome means is we have two chromosomes with exactly the same genes on them. So you and I each have two chromosomes that code for eye color, and we have two chromosomes that code for hair color. We receive one of those chromosomes from our mom, right, and one of those chromosomes from our dad. So really, our genome consists of considerable redundancy. We have two copies of every gene inside our body. And that's a great thing, because if there is a faulty copy of a gene, we potentially have a backup. But there is an exception to that rule, and that exception is the sex chromosome. What I want to do really quickly is dim the lights so that you can see a karyotype. Maybe I'll just try and dim the front lights. That's what this little black square is right here. This is each one of our 23 pairs of homologous chromosomes. And the way that you read a karyotype is, all these chromosomes have been dyed so that specific gene regions glow a different color under fluorescence. And two chromosomes that have the same genes are going to glow the same color. And so you can see like this pair right here, the tips have this pinkish color and then yellow and green and red and all that. So one of those chromosomes was received from mom, and one of those chromosomes was received from dad. Now, the sex chromosomes are right down here. That's the 23rd pair. This individual has an X chromosome and a Y chromosome. So that individual is what, male or female? Male, yeah. So males have a non-homologous pair of sex chromosomes. Females have a homologous pair, right? You have two X chromosomes, which is rad. Have you guys ever noticed that with some types of cognitive issues, they are more common among males than they are among females? Like mental handicaps are more common among males than females. Have y'all ever made that observation? It's true. And one of the reasons is we've discovered that many of the genes on the X chromosome code for cognitive processes. And men have one shot, right? We have one copy of all of those genes. If something goes wrong, we don't have a homologous pair on the Y chromosome. So if there is some sort of mutation, then cognitive processes are inherently inhibited. Females, on the other hand, you have a homologous pair. So that if that gene product that's being coded for by this particular gene on that chromosome is messed up, you have the chromosome you got from dad, for example, that has the correct gene, and you can still produce that gene product. Now, that is absolutely not the case with things like trisomy 21, Down syndrome, or trisomy 23. Those are totally different. And there should be a relatively equitable distribution of those types of disorders between males and females. So all somatic cells are called diploid, which means that they have both copies of each chromosome. They have the full complement, 46 chromosomes. When you go through mitosis, you start off with a cell that has 46 chromosomes, and you end up with two cells that each have 46 chromosomes. But when we go through the process of making gametes, like a sperm cell or an egg cell, then those cells end up being haploid. They have half the number of chromosomes. So a sperm cell carries 23 chromosomes, and an egg cell carries 23 chromosomes. Why would we want that? Why would we want haploid gametes? What do you think? Oh, exactly right. Think about what would happen if gametes went through mitosis, and an egg cell and a sperm cell each had 46 chromosomes. When fertilization occurs, how many chromosomes are you going to end up with? 92, right? And then the next generation is going to have 184. And then there is like this exponential increase in the number of chromosomes across each generation. But through meiosis and by producing haploid cells, if a sperm cell that has 23 chromosomes fertilizes an egg cell that also has 23 chromosomes, then we're going to get a zygote that has 46 chromosomes, right? So the process of meiosis is important for a bunch of reasons, one of which is constraining chromosome numbers across generations. But the important point here is gametes are haploid. They have half the number of chromosomes. Okay, now I'm sure all of you have seen diagrams of chromosomes drawn like this. They look kind of like an X, right? It's kind of what a chromosome looks like. This is a chromosome after the S phase. So this is a duplicated or a replicated chromosome. This chromosome consists of two sister chromatids. So the chromatids are an arm and a leg. One chromatid consists of an arm and a leg. So this is a sister chromatid. As is this. Okay, so what's going to happen eventually is when we go through mitosis and we're splitting DNA into two daughter cells, we're going to take those chromatids and we're going to pull them apart so that one chromatid goes into one cell and the other chromatid goes into another cell. All right? In lab, when we do the cell cycle, you guys are going to be able to see some chromosomes. Excuse me. Sister chromatids are held together by this structure right here called the centromere. It's just that constriction point at the middle of the chromosome. Okay. So chromosomes have some particular organization. And it's important that they have this organization because the DNA molecule is like a really long, really delicate sort of molecule. It's estimated that within each one of our cells, there is like 2 meters of DNA, which is over 6 feet of DNA. So it's incredibly thin and incredibly long. And so what we have to do is we have to make that structure a little bit more robust. And the way that we make that structure a little bit more robust is by twisting it. So if I was to take this string and I was to twist it together, right, a whole bunch of times, then eventually what's going to happen is that's going to be a little bit more strong because you have 2 strands that are intertwined. But what's even cooler is if you twist it up enough and then you relax that strand a little bit, it's going to coil in on itself. And so when we're making chromosomes that we actually want to start pulling around the cell, and we want to condense them, this is how we do it. We just take DNA and we coil it up really, really tightly on some scaffolding proteins and some associated what are called histone proteins, and we just make a really, really tight bundled up structure that looks a little bit like that. And then we're able to pull that around the cell without breaking the DNA molecule. When it's not wound up, it's called chromatin. And that's an important phase of our DNA. When cells are in arrested development, when they're in between cell cycles, some of our DNA is in the chromatin state. And it's an important state because that DNA is accessible to cellular machinery, so that when we need to make a protein, we can get at the blueprint for that protein inside the DNA. If we start twisting it up really tightly, we make that region of our DNA inaccessible to cellular machinery. Okay, so when we want to start moving our DNA around, what we do is we coil it up, and we coil it around some proteins called histones. And histones are little globular proteins. They look, well, you can't really see the diagram very well, but they look like just a little ball. And histones inherently have a positive charge, which is important because DNA carries a negative charge because of that large phosphate group in the backbone. And so what we'll do is we'll take our DNA and we'll wrap it around a histone. And then we can take clusters of histones that are called nucleosomes and twist them together. So a nucleosome consists of 8 histones and some DNA that's wrapped around those 8 histones. And we get those things to stick together by twisting up our DNA, pulling it in really, really tightly. We can then start taking these 8 histones in the DNA, those nucleosomes, and twisting them together and creating something called a solenoid. So this is what a solenoid looks like. It's just a whole bunch of nucleosomes that are stuck together. We can then take those solenoids and we can twist them together to make what are called looped regions or looped domains. And then we take a whole bunch of looped domains and we attach them to a scaffolding protein to make something called a rosette. And it actually looks like a floral structure. So if I was to diagram that on the board a little bit more large, a little bit larger.
Did you say a bunch of the stones just like?
A bunch of histones stuck together is called a solenoid. So our rosette would look something like this. We're running up the middle of a whole bunch of rosettes. There's something called a scaffolding protein. And that's what this is. And then what we do is we take our DNA, these large rosettes, and we just attach them all to the scaffolding protein like this. And you get rosettes like this distributed across the length of the scaffolding protein. Now, each one of those rosettes consists of a whole bunch of solenoids. And those solenoids are just collections of nucleosomes. And those nucleosomes are just histones plus DNA.
So the rosettes are connected to a scaffolding?
Mm-hmm. Scaffolding protein.
I'm sorry, histones are connected to scaffolding?
So think about it in terms of hierarchy.
Yeah.
You get a histone, which is just a globular protein, and you wrap some DNA around it. Then you take a whole bunch of histones and you stick them together to make a nucleosome.
Okay.
Then you get a whole bunch of nucleosomes and you stick them together to make a solenoid.
Okay.
And then you